stuff for you. It's called DNAcid. It's really strong, so be careful not to take it all at once, okay?
Relax, bro. I have this under control.
I know.
Yeah. Oh. What the? Whoa! What is happening? Ah,
Hello. You were warned about not taking too much. Now you're going to learn all about the structural composition of what you just took, the oxyribonucleic acid. In 1953, James Watson and Francis Crick became the first to discover DNA. Since then, we've learned many things about this type of nucleic acid. DNA stands for deoxyribonucleic acid, and it is made up of two strands, the leading and the lagging, and also is made up of four types of nucleotides: adenine, guanine, thymine, and cytosine. Adenine and guanine have what's called a double ring structure, while thymine and cytosine have a single ring structure.
what are nucleotides made of?
All nucleotides are made up of three things: a sugar, a phosphate group, and a nitrogenous base.
How do they bond?
Nucleotides form hydrogen bonds with one another. However, not all of them can bond together. Adenine will always bond with thymine, and according to Chagres' rule, there will always be an equal amount of adenine and thymine in any given DNA sequence. Adenine and thymine form two hydrogen bonds with each other. Likewise, cytosine and guanine will form bonds exclusively with each other and will also follow Chagres' rule, meaning that there will always be an equal amount of guanine and cytosine in any given DNA sequence. Similarly to adenine and thymine, cytosine and guanine have three hydrogen bonds between them.
Oh god, I wish I have more of that pills. That will make me pass biology, but now, I suck. Who are you?
We know what you want. We know what you mean.
No, not again.
Welcome back, boy. With this pill, we're going to explain DNA replication, transcription, and translation. DNA replication is one of the most important processes for the cell. It involves four major enzymes: primase, ligase, helicase, and DNA polymerase. First, the DNA is unzipped by the helicase. A helicase breaks the hydrogen bonds between the nucleotides. DNA polymerase is the enzyme that bonds the nucleotides. It is led by the ligase in the lagging strand and by primase in the leading strand. Although they seem similar, these two strands function completely different. The leading strand goes from 3' to 5', and nucleotides only bond on the 3' side of a strand. The leading strand is bonding continuously. The lagging strand, however, is different. It goes from 5' to 3', which means it can bond continuously. The lagging strand is bound in a section called Okazaki fragments, which are sets of bonded nucleotides on the 3' side of the strands, which are then stitched together by ligase.
This is one of the most important processes during the cell cycle. Do you know how essential the proteins our bodies create are? Well, that is thanks to DNA. It begins with transcription. The promoter is where RNA polymerase binds with the DNA. The DNA is unzipped and a strand of it is copied into an mRNA. When the RNA polymerase reaches a point called the terminator, it detaches from the DNA. which then zips back up. The difference between RNA and DNA is that RNA has uracil instead of thymine as a nucleotide. Also, that RNA has only one strand. When the mRNA is floating in the nucleus, it goes through intron splicing. The junk parts are introns and they are removed. The remaining parts are the exons, which get stitched together. After this process, the mRNA strand exits the nucleus through a nuclear pore.
For mRNA to create a full protein, it needs to go through translation. This process starts with the mRNA strand getting binded with a small and large ribosomal subunit which has three sections, the E, P, and A. This molecule will attach to the initiator codon of the mRNA, which is AUG, and will bind in the P section of the large ribosomal subunit. Then, tRNA molecules continuously bind their anticodon to a codon in the A site, while the amino acids attached to the tRNA in the P site are passed on and bonded with the amino acids in the tRNA molecule located in the A site. Once this is complete, the tRNA molecules move to their left, and the tRNA molecule is released, while a bigger polypeptide is formed. The process continues until the stop codon is reached and a release factor is binded to it. The stop codons are UAA, UGA, and UAG. When this happens, the polypeptide that was formed gets released, completing translation. The polypeptide that was released can be used later in any part of the cell.